most people may still be unfamiliar with this subject matter. And, when I was learning about viruses in medical school, of course, I was taught, everything that you've been taught. But what was interesting is that they never really went into the original experiments that discovered the viruses or showed that they could make people sick. In fact, they never even talked about the research with respect to contagion. And it wasn't until, the COVID pandemic that I really examined the scientific experiments that were done to, come up with those ideas and give them, scientific validation. And what I found is that they actually didn't find the evidence to support those ideas of contagion and of viruses causing disease. There have been over 100 published studies, and these include some animal studies in horses and cows, certainly, because animals do, get flu-like illnesses in a seasonal pattern just like humans do. But the majority of these over 100 studies were done on humans, and they're all documented, by the way, in a book called Can You Catch a Cold by Daniel Roides, which is available on major platforms like Amazon. And it turns out that none of those studies, and they were all done in a scientific way with a control group, where they tried to take, secretions from people with colds and flu, even the Spanish flu, and expose healthy people to them. They could not reproduce the flu or colds in any of these experiments. So over 100 experiments essentially failed to prove that we actually have contagion of colds and flu through these secretions that are allegedly contain infectious viruses. Now, I'll bet that many of the viewers aren't aware that scientists who allege to discover viruses have never ever in the history of this field, purified a virus in a test tube where you could examine that virus that they, extracted from some organism who was sick and tell what it's made of and tell what the genetic sequences are. They've never even attempted actually to do that. Now, early on in the field of virology, they did attempt that. They were never able to do it, and they just came up with a method that simulates the whole experience. So the only way that they say a genetic sequence is related to a virus is by taking a mixture of animal or human tissue and fluid, and then just basically based on circular reasoning saying there must be a virus in there making that animal sick. And then they look at all the genetic material in that sample, and it's got, human cells in there of various types, bacterial cells, fungal cells, yeast cells. Now, they may filter those things out, but they secrete genetic material into the soup that's with those cells that stays in there. And then they analyze all of the genetic material in there, not knowing what piece is from what organism. And they put it into a computer program and basically do some algorithms on it, and then at the end just say, here's 1 million possible virus genomes among these sequences, and then they pick one of those out of 1 million and say that's the virus sequence. And they've never traced it back to find, is that sequence actually in a virus, because they couldn't do that because they never ever had a virus in a test tube where they could do that kind of an experiment. So they basically have, simulated in a computer model a virus that has never been proven to be in reality. Then they validate a test using synthetic genetic material because they don't have the real thing. And they say, oh yeah, this test is valid. It measures this genetic sequence. The only problem is we have no idea where that genetic sequence comes from or if it even really exists in nature. Now, the test that they used is a PCR, or polymerase chain reaction test, right? And many of us learned what that is during the COVID era, because that was one of the major types of tests used then. And these tests, right, were not given full approval by the government regulatory agency, but they were given an emergency use authorization, which means they could be sold without the company making them having to prove that they actually work. With any diagnostic test, the first step that you have to do to show that it actually diagnoses something is called a validation study. And this is where you compare it with the gold standard to show that it measures what the gold standard measures. And so if we were to develop a pregnancy test, for example, the gold standard is that a baby comes out at the end of the pregnancy. And that's, pretty objective, right? We know that that is the gold standard. And so we can do a pregnancy test early on and then wait the rest of the pregnancy and say, does a baby come out? And we can compare the results. And if they agree 90% of the time, that's pretty good. And I knew about this trick because the CDC also did this trick during COVID. They didn't use a gold standard, because the gold standard would be comparing it to actually finding a virus in an animal by an electron microscopy of a biopsy of the animal's tissue, for example, and then comparing the test to that. But that was not what was done at all. Instead, what they did was they took a sequence of genetic material that they said was from the avian flu, and they made it synthetically in a laboratory. Actually, there's machines that they use for this, like the DNA and RNA synthesizing machines, where you just add a bunch of chemicals to it, type in the sequence in the computer interface, and it takes hours or days, and then it just spits out this particular sequence. And they compared that as the gold standard. So something that's completely made up.